Hi, my talk today is going to be on metabolomics and exposomics, new tools for the health care and environmental policy and disease prevention, diagnosis, and treatment. So these are my disclosures. And for references, convenient compilation is a monograph of 26 papers I put together that's available at our website link. It's under number 11, reading. And you can just download those papers. They cover 50 years of research. Also, you can go directly to the journal Mitochondrial, and 4 of the top 10 papers, the most downloaded papers, are on mitochondria, autism, the cell danger response, and healing. So a lot of our perspective comes from this statement by John Dryden that self-defense is nature's oldest law. All of us have inherited the genes from our ancestors that allowed them to survive every famine, every cold winter, every microbial infection or pandemic until at least the birth of their first child. Another interesting thing is that it's embodied in a statement made by Nelson Mandela that when a deep injury is done to us, we never heal until we forgive. So a friend of mine, Keith March, asked the question, how does a cell decide when to forgive and to begin to heal? And my response was, a cell will only redirect resources away from cellular defense toward healing when it receives the signals that say the future is safe. So the corollary to that is regeneration and healing cannot proceed until the persistent triggers of the cell danger response are removed. So why do people become chronically ill? It's because they encounter a perfect storm of dozens of environmental and genetic factors that produce or persistently reactivate injury and then block the healing cycle. And all chronic illnesses represent a failure of our ancestral genes and the inherited epigenetics to adequately manage current environmental conditions experienced by a patient. So to summarize bluntly, people are sick because they can't get better, because they can't heal. So what are the organizing principles for my talk? Well, first is injury is universal. Living cells try to heal themselves. This is a biological process, not an engineering process that we can force. Chronic illnesses are the result of a mismatch between the conditions experienced by our ancestors and the current gene environment and developmental timing interactions that occur during injury and exposure. So the gene by environment by timing, the GET principle is whether people get healthy or get sick is determined by ecogenetics, the interaction between our ecosystem and our genetics. So metabolism is the real-time interaction of genes and environment. So metabolism lives in the moment. And this other principle that's an important concept is the brain ultimately controls peripheral metabolism. So the take-home messages for this talk are, one, that our pathogenesis is actually separate from and does not equal salutogenesis, or the process of, otherwise process of injury does not equal the process of healing. And the cell danger response and mitochondria coordinate both injury and healing. And the metabolomics can be used for pre-symptomatic risk assessment, triage, diagnosis, and prognosis. So here's another cartoon. It basically illustrates that we can't see what we can't imagine. Our training creates subliminal limits. These are unrecognized horizons in our vision that put blinders on so that we can't see the full extent of the physical and biological reality that surrounds us. So new tools and models are needed to extend our scientific vision. And I'll talk about how sometimes all it takes to discover a new world is a new lens, like von Leeuwenhoek's discovery of protozoa in a drop of blood, the antimolecules, excuse me, not in a drop of blood, a drop of rainwater, using his microscope. To understand a chronic illness, you need a paradigm shift. And for this, what we've adopted is this categorical difference between the understanding and treatment of acute disease and chronic disease. And we've called The medicine that has evolved over the last 5,000 years of recorded history as book one of medicine, and book two is needed in order to address the causes and treatment of chronic illness that lasts about more than 6 months. So what's another perspective? So if we talk about a healthy biology like this house here that might become under fire, so that's the process of pathogenesis, and we look for microbes, a source of infection, trauma, toxin, where we can treat symptoms as well. And then we can use antibiotics or statins to lower cholesterol, insulin to control glucose and put out the fire. But that leaves us with a broken house, a burned down house. Unfortunately, a lot of medicine currently just stops here and assumes that the body will heal itself. That may have been something that was, a more reliable outcome in years past, but I'll make an argument toward the end of this talk that things have changed considerably, and the environment that we live in is no longer as conducive to healing. So the rebuilding process requires active healing, and that's what I call this process of salutogenesis from the Roman goddess Salus, the goddess of health. And that active healing requires new energy and resources, biological energy and resources. All right, so here's the Roman goddess of health, Salus, and this slide just makes the point that we go from a state of health to a state of chronic illness through a variety of different pathways that can be trauma, a broken leg, or poor nutrition, or abnormalities in the microbiome, things we do to ourselves that have to do with pollution and tobacco. Anyway, we find ourselves in this repeating loop of chronic illness, and it turns out to get back to health requires a different process that is not the rewinding of the tape of the process of pathogenesis. And this different process has a beginning, middle, and end that are supported by and absolutely require three different mitochondrial transformations, what we call M1 mitochondria that are pro-inflammatory, M0 mitochondria that allow for cellular replacement and proliferation, and M2 mitochondria that are anti-inflammatory. So the first book of medicine really focused on the process of pathogenesis and relied on natural healing once the damaging cause of damage was removed. But nowadays, it turns out that we can remove those sources of damage and still not achieve recovery. So for example, insulin has never resulted in a cure of diabetes, and statins have never resulted in a cure of dyslipidemia. So in salutogenesis, in the second book of medicine, we're using this inherited biology to identify salogens that can remove blocks and facilitate a return to health. So when I gave this talk at the National Institutes of Health in April of 2019, the conference organizer came up to me and said, "Great talk, Dr. Naviaux, but I hope you are wrong." I said, "Why is that?" And he said, "Well, because if you're right, then for about the last 70 years, NIH has been focusing on support for just one limb of a two-limb problem, okay? The one limb being pathogenesis and the second limb being salutogenesis." Okay, so many silos of chronic illness and disabilities are connected by a common root pathology. So I'm going to talk to you in this talk of our work in primary mitochondrial disease, autism, ME/CFS, Gulf War, PTSD, Lyme, PANDAS, suicidal ideation, ALS, Parkinson, et cetera, even healthy aging and exercise. So we've studied the metabolomics of each of these, or are beginning to study in the cases of ALS, Parkinson, and Alzheimer's. And the thing that has become clear is that there is a connection between all of these that is shared. So the philosophy of biomedical research in the West has largely been to build deep silos of knowledge that allow us to say how primary mitochondrial disease is different from autism, different from ME/CFS, is different from Gulf War. But in fact, there's this hidden connection of a root biology of the persistent cell danger response that creates blocks that prevent each of these different disorders from returning to health. So things that make chronic diseases the same are more important for healing than their differences. So in other words, unification is more important than division, which is a sentiment that is important for, politics in America as well. So mitochondrial function changes and must change in order to fight infection, to learn to heal. So over on the left here, we have a picture of cells with an interconnected network of mitochondria that are stained red. Okay, healthy cells have this interconnected network. As soon as there's an injury, that network becomes fragmented. So you can see these little blebs here. This is what we call the spaghetti to meatball transformation of mitochondria. And that initiates pro-inflammatory stages of the first stage of cell danger response, what we call CDR1. But that's absolutely necessary to initiate the healing process, the healing cycle we call it, and to return cells to health and fitness. And when that happens, the mitochondrial network is also reconnected. However, what if there are blocks for a variety of different reasons to that healing process, and that the cells initiate a repeating loop of efforts to try to heal, but unsuccessly? But that can lead to chronic illness in adults. That's 60% of adults have at least one chronic illness, and in children, in the U.S., about 40% of children are living with a chronic illness now. And 35 years ago, when I was in medical school, this number was 5%. So what has led us in this direction of the cell danger response? So we've looked at over a dozen different studies in different disease cohorts of autism, ME/CFS, Gulf War, et cetera, down through this list. And it's led us to an understanding of a conserved biology. So you can envision this like a Venn diagram where there are metabolites and pathways that are present in ME/CFS and unique to ME/CFS, others that are unique to suicidal ideation or traumatic brain injury, Gulf War illness. But there's also a shared biology that is the same for all of them, and that's the cell danger response. And when we look at the pathways that actually are regulated in order to help the cell heal, it involves purines and pyrimidines and phospholipids and fatty acids, acylcarnitines and sphingolipids, and a variety, a number of different things that are familiar to anybody who's spent some time in the biochemistry of chronic illness. But this comes up over and over and over again in studying, over a dozen different diseases. So what is the cell danger response? So here's a cartoon of a cell, and maybe an archetypal stress might be a viral infection. And what that does is lead to a cascade of cellular responses that involve first or very early in the process a conversion of filamentous mitochondria to fragmented mitochondria. And when that happens, there's less oxygen that is consumed by mitochondria. And because the capillaries around the cell are still providing oxygen, it leads to an increase in dissolved oxygen concentration that then begins to actually push macromolecule synthesis toward smaller molecular weight building blocks. There's a release of reactive oxygen species. There's a demethylation that occurs that activates a more promiscuous gene expression, including the release of endogenous retroviruses and the release of eicosanoids, synthesis of endocannabinoids, and a production of what we call sickness behavior. So all of these things work together. They're not in isolation at all. So here's another set of calculations that are necessary to understand how we interact with our environment. It turns out just the process of breathing every day, we take in 15 kilograms of air in a day. We take in also about 3 kilograms of water and about 1 kilogram of food. So this ultimately leads to this inference that when you consider toxin exposure on a parts per million basis, that air quality is 15 times more important than food quality, and 5 times more important than water quality. Okay. And I've looked at things like noise and what the median level in decibels is, and electromagnetic fields. And just to give you an example, we're on average exposed to just a few, 1 or 2 milligauss a day if you're a computer worker, or 8 to 10 if you're an arc welder. But by comparison, an MRI is a 1.5 Tesla machine, which is 15 million milligauss. Okay, I won't go into the rest, but it's there for your reference. So in the first book of medicine, health is interrupted through injury. We apply the principles of first aid to airway, breathing, circulation, as well as antibiotics, symptomatic treatment. And we rely subconsciously on this black box to occur. That black box is healing. And if it works, we get healthy. But if it doesn't work, there's failure, and it leads to chronic illness. So if that continues to go this way, then we end up with about 40% of our children and 60% of adults in this repeating loop of chronic illness, unable to get back to health. So what we propose is that we try to unpack the molecular features of the healing cycle, try to understand this as a programmed ontogenetic sequence that has a beginning, middle, and end. And maybe by understanding this, we can unblock the healing cycle and lead more reliably to health from chronic illness. So this is a universal process. The cell danger response is something that all eukaryotic cells on the planet had to evolve. And this is an example of Arabidopsis, a little mustard plant that's been modified so that it fluoresces when intracellular calcium is released. And so this is a little cricket bite over on the right-hand side. It turns out that if you add either glutamate or ATP, extracellular ATP, then the message of injury is propagated to the rest of the plant within two minutes. So this has also been done in venous flytrap. That's also been made recombinant with a calcium GFP protein. And this is a case that illustrates this principle that eating can be dangerous. So over on the left, we have a venous flytrap that has been stimulated, and there's very rapid, about 10 times more rapid distribution of calcium into the plant. And then over on the right, what we have is when this petal of the leaf is cut off, that single signal of danger is passed by calcium signaling through the veins of the plants very rapidly. I'll show that one more time. Kind of like a primitive nervous system, except it was working through the actual vascular system of the plant. Okay. So there are 3 kinds of mitochondria that are absolutely required to heal from any injury. We call these the M1, M0, and M2 mitochondria. So normally we spend our days moving in this healthy developmental cycle with wakeful activity and nutrient intake. And there's a little bit of ATP that's released from every cell. And that ATP is then metabolized to adenosine that's necessary for restorative sleep, and the cycle continues and for adaptation. But if there's too much stress or injury, then the cycle takes on another path that starts off with initially a containment phase that activates innate immunity and inflammation. But that is absolutely dependent on mitochondria undergoing an M2 to M1, basically spaghetti to meatball transformation. And in this video micrograph off to the right, mitochondria in a HeLa cell have been stressed. And then within 5 minutes, they alter their configuration and fragment into this pro-inflammatory morphology. Once the damage is contained and the microbe eliminated, then the cells lost in that originally inflammatory stages have to be replaced through cellular proliferation in CDR2. And then once the cells are replaced, those newly born cells have to take on the gene expression characteristics and metabolic characteristics of the neighboring cells as they do in CDR3, as mitochondrial oxidative phosphorylation reinitiates and the mitochondrial network is reestablished. So again, I'm just emphasizing that this involves a mitochondrial transformation that is absolutely normal and absolutely necessary. So fragmented mitochondria that are pro-inflammatory are M1, mitochondria that adopt Warburg metabolism or aerobic glycolysis are called M0 mitochondria that support cell growth. And then M2 mitochondria put the brakes on cell growth and allow for full differentiation. So chronic diseases, we believe, results in large part because of blocks in the healing cycle. And autism and ME/CFS result in blocks largely at checkpoint 3. So we envision the study of metabolomics and exposomics like taking a drop of water from an ocean ecosystem, a coral ecosystem, a river ecosystem, and looking at the little microcosm of chemicals through mass spectrometry. And so we do that through metabolomics in one kind of mass spec and exposomic in another kind of mass spec. And at UCSD, we have a method that allows us to look at nearly 700 natural chemicals, phospholipids, fatty acids, purines, sphingomyelins, from about 65 different biochemical pathways. We can also look at a total of about 1,200 environmental toxicants and chemicals, including pesticides and flame retardants, phthalates, persistent organic pollutants, mycotoxins, food additives, flame retardants, etc. So nearly 2,000 chemicals we can measure. So when we've applied the metabolomics methods to different chronic disease cohorts, including autism, PTSD, Gulf War illness, et cetera, we find that our accuracy in using accuracy in diagnosis is basically 85 to 95%. And in some experimental models, it can be as high as 100% in a laboratory model of Dower, which is a hypometabolic state that allows for long-term persistence in the face of environmental hardship. That we believe is very similar to ME/CFS. But we can also use metabolomics to predict future events. And it turns out that if you draw blood at time 0 in patients that have had a history of major depressive disorder, and then follow them, so these are patients who are healthy and are in remission, but then follow them over time in females and in males to those who have recurrence of depression. We can predict with 90% accuracy in the females and 99% accuracy in the male which individuals will have a relapse. And sometimes we can identify specific metabolites like a reduction in methylcysteine that increases relapse in females and a reduction in beta-carotene that increases the risk of relapse in males with major depressive disorder, with a history of major depressive disorder. So now I'm going to turn to our study of exposomics, which we look at as the footprints of a changing chemosphere, and then ask the question, what happens when you measure 1,000 man-made chemicals in the blood? Well, when we did that in a cohort of 4 to 6-year-olds with autism and typically developing controls, we found 27 chemicals that were present in at least 75%. So 75 to 100% of the samples had these 27 chemicals. But only 3 of them were statistically different between autism and healthy controlled children. And what was even more interesting, that the differences were that the children with autism had a decreased concentration of perfluoroalkyl acid, which is a Teflon, and dibenzyl-acridine, which is a car exhaust component and a carcinogen. And we found that a common food preservative, terse butyl hydroquinone, was present in 19 of 31 children with autism, but 20 of 22. So the overall rate of finding this in children with autism was, only 2/3 the amount in children who are typically developing. But maybe the bigger message is actually this sea of environmental chemicals that we're all swimming in that include pyrethroid insecticides, hydraulic fluid components, fungicides, dyes, carcinogens, endocrine disruptors, many, many different kinds of Teflons, herbicides, food sweeteners, etc. So PFNA is that perfluorononoic acid. You can see that it's lower in autism than it was in TD. It's also the PFNA, this is its structure off on the right, this perfluorocarbon that was found to be at highest concentrations of 100 parts per billion in Atlantic dolphins and up to 400 parts per billion in polar bears. Okay, so the blood levels, yes, well, let me just say that the new municipal water standards in New Jersey require PFNA concentrations to be below 13 parts per trillion. Okay, that means that the blood levels that we're finding in dolphins and bears are 10,000 times higher than the safe amounts in water. And dibenzacridine was lower also in ASD than TD. This is what it looks like. It's a polyaromatic hydrocarbon. And so this led us to a corollary question: could the restricted food choices seen in over 80% of children with autism be driven by autonomic behaviors that were evolved to decrease the exposure to foodborne chemicals, kind of in keeping with that original principle I introduced that eating can be dangerous, that trigger the cell danger response and anxiety, and this butterfly response that's highly a negative and aversive autonomic and metabolic response that might be due to an increase in purines like ATP and even xanthine. So in recent studies, it's been shown that xanthine release during times of stress is absolutely necessary for producing anxiety-associated behaviors like the avoidance of open fields. Okay, so the closing question is, what if the chemicals found in the exposome of children with autism are not specific at all, but instead act as an invisible rising tide, the tip of an iceberg of thousands of chemicals that are in this sea that we live in, the sea of a changing chemosphere, that actually affect everyone's health, not just children with autism, and is ultimately responsible for many chronic illnesses like ME/CFS, ALS, and autoimmunity. So I'll end there and give thanks to our financial supporters, including the Christini Fund and over 2,000 grassroots and anonymous donations, and the Seeking Health Fund and Optimizing Lives program of Dr. Lynch, and many others. So thank you.